Hello, my project is predicting patient progression using gene expression analysis, applying machine learning to high-dimensional biological data. So maybe I should start with the motivation and big picture. And as you can see in the picture, beside the university courses, I also learned about Chinese feng shui to predict something by using somebody's birth time. So the most frequent questions I got asked is their health issues, but time flies by. I start to realize that I'm also studying in data science, and can I predict the health issue also by using another method, data science method, uh, besides the Chinese feng shui, and so maybe predicting outcomes like progression earlier allows doctors to identify high-risk patients because I also learned about informatics. So this enables more personalized treatment plans, potentially improving patients' care and prognosis. And furthermore, understanding which genes are linked to the progression can guide research into new therapies and diagnostic markers. And finally, maybe I can prepare it as a start of learning machine learning. Okay. The main idea is there are actually some diseases highly related to the gene expression in human RNA sequences. If some of them could be predicted in bioinformatics sense, like some cancers caused by DNA and RNA genes, etc., that means we can use machine learning to play a crucial role in it. By analyzing the gene expression level table, maybe we can find some genes are highly associated with a certain disease. If it's applicable, perhaps it could be a major reference for doctors. So very luckily, I find some data sets on Kaggle, and it is about the neuroblastoma, which is a disease frequently occurred in kids. So the data sets contain main two data sets. One of them is the expression level matrices. The row are the different genes in RNA sequences, and the column is the different cases of patients, and also the another patient info TSV dataset, which contains the age, sex, and etc. So the first thing I need to do is to merge two different datasets. I need to match the patient info and the gene expression level into one single dataset because they have different row and columns, so I need to transpose one of them. And second, I need to define the target variable and features. I need to predict something. So the variable is the progression I choose. The disease progression is the critical clinical endpoint. By knowing whether a patient condition is likely um, to worsen is extremely valuable for doctors and patients. It directly impacts prognosis and treatment decisions. And also the features, as you can see, is very easy to observe, is the number in the matrix, the expression level to each genes. But the question is, when I visualize the dimensionality of the data set, it comes to my attention that the dimension is very, very high. You can see the gene features, the abundance of gene features, but very few samples. This is the case of the P large than N. Then the P stands for the features and N stands for the sample. In machine learning, there's a specific model to tackle such situation, which is lasso regression. So lasso is a type of regression model well suited for high-dimensional data. It performs regularization, which penalizes model complexity, and crucially, it can shrink the coefficients of irrelevant genes to exactly zero, effectively removing them from the model. So as a data scientist, I'm very familiar with the standard linear regression equation. But in the lasso equation, there's a extra penalized function. In each learning, as you can see the lasso coefficient paths, the lines represent the different genes. Finally, after many time training, as you can see, most of them become zero, which means in such penalized, some of them, most of them are actually contribute very less to the model. So one important question you may ask me is that how to choose that penalty term. In here, I use tenfold cross validation. It can help us to find the best penalty level. The plot shows the optimal lambda that maximize the AUC. As you can see, the peak is between the 9 and 15. I think that is 10 or 11. So the most important is to observe the evaluation. The ROC graph shows the AUC is 0.8217, which is very good. And the confusion matrix, there are only one type 1 error and only 13 type 2 error. And the overall accuracy is 0.72, which is generally very good in this certain situation. The sample case is very small. So the result visualization shows us that the lasso features important graph shows that these are the genes that model things that are highly associated with the occurrence of the neuroblastoma. But well, it is hard for me to show the all genes to do the comparison. As you can see, you only need to know is that there's so many, around 20,000. That's why lasso regression is very important. Just few among 20,000 genes, only around 1, 2, 3, 4, 10 of them are highly associated with the disease. But we are data scientists, right? That is not the end. Maybe we should try another method to do the comparison, like compare which model is good. Maybe we can try the we'll call rank some tests that is selections based on their individual association with the outcome, and building model by using only top rank genes to split it into to try with different size of subsets like 10, 20, 15, and 100. And the graph is very hard to observe, and I also print it with RStudio. And you can see the top 20 genes returns the highest AUC, but it is still less than lasso regression. So maybe lasso regression is the best model in such cases. So by doing this project, I learned so many things and comes out that maybe my idea is applicable. Maybe, but there's a lot of things I can do better. There's a further step I should do. This alignment, the local alignment and global alignment, but I lack of RNA sequence data set, and most important, a powerful computer. And also the sample size is a main issue. The because the sample size is very small, so it's not accurate as I would expect. Also, the data is from a single cohort. Maybe it lacks generalizability. And most important, the cause to a certain disease to be occurred can be very complicated, so it may be very subjective.